Okay, so sorry about all of that. It looks like we're set to go. Lauren, thank goodness, is here to help me and is going to advance my slides for me. So since we've gotten off to a little bit of a late start when I practiced this, it was about an hour, so we'll just get right into it. I'm super excited to be able to talk to you about epilepsy genetics. This is going to build on some of the discussions that Katie Angioni, our genetic counselor, and myself have given at a couple of these in prior years. So let's get started. Next slide, please. So these are the topics that we're going to cover today. Hopefully we'll get to all of them, but some kind of lingering questions and new discoveries that I think are going to kind of transform the field of epilepsy genetics. Next slide. All right, so we'll start with structurally mediated epilepsy and our discussion of mosaicism. So structurally mediated epilepsy is one of the most common causes of seizures in people. There have been a few estimates of incidence, about 20 cases per 100,000, and then a study conducted at the Mayo Clinic in Minnesota between 1980 and 2004 estimated it to be about 27% of children with epilepsy. Structural malformations that can cause epilepsy range in severity from major malformations that can affect much of the whole brain, hemimegalencephaly, lissencephaly, polymicrogyria, which can be small locations or bilateral or large parts of the brain, to other small lesions such as focal cortical dysplasia that may or may not be visible on MRI. Frequently, this type of epilepsy is actually quite treatment resistant and intractable and accounts for a large portion of those treatment resistant patients. Unfortunately, we don't understand a lot about how these types of malformations form and how to manage them. And so many of these patients actually do progress to surgical interventions. And a lot of the success of those interventions depends on how well we can figure out how extensive the malformation is and how extensively we resect it based on other limitations. Next slide. So the concept of mosaicism is the idea that more than one genetically different line or set of cells exist within a single body. And we naturally are mosaics, especially in our brain, because our brain cells start all around a central canal, which becomes our ventricular system that contains the fluid that bathes and nurtures the brain. And those cells start there in a collection, and they begin to migrate out. They organize themselves into the ridges and folds that we think of as the shape of the brain. And in doing that, they also rapidly divide during that process. And as they divide, they may not copy over our genetic material with 100% fidelity, meaning that there may be slight changes in some of those cell lines as they divide and create our brain. The extent and location of that mosaicism and those different cell lines depends upon when that happens in the course of fetal development. So this is a great pictorial slide from a paper by Ann Peduri, which kind of puts into a picture a concept of this type of genetic variation. So on the left, you see that if there is a mutation or a variation in a gene in the parent, that is inherited and it affects all the cells in the child. And that is called a germline mutation. However, if the egg and the sperm meet, they form the zygote, the beginnings of the new baby and the fetus, and a change happens subsequently as this genetic material is being copied over. That change is reflected in only some of the cells of the baby. And this being what ends up becoming our brain cells. So you can see in this particular condition, it affects not just the brain, but other parts of the body as well. In this particular slide, which is hemimegalencephaly, you see that the change happens a little bit further on in cell division and in the development of the fetus, such that it is only contained in some cells of the brain and no other body tissues. Unfortunately, our traditional genetic testing mechanisms have not really been able to pick up mosaicism very easily, and that is changing. Our detection, our mechanisms are getting to be much more detailed and effective in picking this up. However, this testing also is dependent upon the tissue that is sampled. So if we do traditional blood testing for a patient who has a mosaic mutation causing their brain malformation, that's somewhere up here, and we may miss the actual tissue in which the gene change is present. In our clinics, typically for children with structural epilepsy, we tend to take cheek swabs, which will get us ectodermal tissue that is closer in cell line to brain tissue. However, we are not actually sampling just the brain, right? And so we may still miss some of these causes of structural epilepsy. Next slide. So this is, again, a growing area of discovery and research. And so I want to present this particular study, which looked at multiple surgical specimens from patients with structurally mediated epilepsy and reviewed the sequencing that had been done on that brain tissue. Of all of the patients, and these patients had focal cortical dysplasia, one of the kind of less prevalent, but meaning less extensive brain malformations, but very common. Of those 307 patients, about 13% of them had an identified genetic variant as the cause of that structural malformation. And they've kind of categorized them by color here. And most of them were in this mTOR gene. And then these other, whoopsies, I lost the presentation. Sarah, did you take over for Lauren? Hold on, let me try again. Oh no, apparently the internet just went out at the hospital. Fantastic. Apparently none of the other presentations have had this issue, so it's just lucky me today. So, oh my goodness. Chelsea, are you there, Chelsea?
I'm here. I'm here.
Could I send you my presentation and maybe you?
Yes, I am also at the hospital.
Oh, shoot, you're not on your own laptop?
No, I'm not on my own laptop. I'm on my own laptop. I'm on a room at South Campus.
Okay. Oh, shoot. Oh, it's campus. Maybe Lauren is at the main one. Is your internet out as well?
Let me see. Hold on. Yes, it appears to be. I'm not connected to the internet. My phone's not connected to the internet.
Okay. I'm going to go upstairs and try to log on.
Oh. Now it is.
Okay. Let me send you my presentation.
Okay. Yeah, I got it. Open. And then let me share my
unmuted.
Let me see if I can share my screen. How about that? Can I do that? Huh. I should figure this out before I go next time. Share screen. There we go. Wait, that's mine. Oh, I gotta close some of my patient stuff. Hold on. Okay, closing Epic. Sorry guys, I just want to be extra safe. Okay, I think I'm sharing this one. Is that working?
Give it a second. This happened with Lauren too. It has to just load.
Has to give it a moment.
There we are. Okay, we were at the slide number 5.
Great.
Wonderful. Okay.
Then I'll go slideshow from current slide. Let me, you can just say next.
Thanks, everyone.
You go next.
Okay, wonderful. Okay, so again, they identified this mTOR gene as containing a lot of the variants, but these other genes were also identified, and those are actually also within the mTOR pathway, which you'll hear about in several other of today's slides. You know, I think these results are a little bit skewed because they really only looked for genes within the mTOR pathway. And so more recent analysis have also identified other genes in different pathways, some of which we actually associate with other epilepsy syndromes, like PCDH19, NF1, so on and so forth. There's also something called the 2-hit hypothesis, meaning that it's possible for people to have both a germline mutation, meaning something inherited that's present in all of their cells, and then a second problem occurs in some brain tissue cells. And this is actually what results in the dysplasia or the malformation, is the combination of those two things together. But they don't have the full presentation of the condition because that one is not sufficient for symptoms to occur. And this has actually been shown in a couple of genes. Tuberous sclerosis, there was a patient reported with a germline mutation in TS, the TSC2, and then a brain tissue mutation resulting in hemimegalencephaly. And that patient did not have any other systemic manifestations of tuberous sclerosis. So no heart rhabdomyomas, retinal findings, skin findings, none of those other things, just a brain malformation from a different mechanism than is usually seen. And this was also seen with STXBP1, which is a known developmental and epileptic encephalopathy gene. Some research has also shown that in the surgical specimens themselves, the highest percentage of the mosaic mutations, so seen in 25 out of 100 cells, was present at the epicenter of where the seizures were detected in intracranial monitoring, and that level of mosaicism decreased gradually as you got further and further away from that epicenter. So at 25, it was 25% of cells here, and then 20, and then 15, and then 10, until you got to normal brain tissue. And so I think this is really exciting because that, you know, kind of opens up the possibility that potentially we would be able to determine the borders of a surgical process for epilepsy by looking at the genetics of the tissue and saying, is this mutation that posed a risk for this patient developing seizures still present in any amount at the end of where we were planning our surgery, or do we need to go further? Next slide. So I think there's a number of clinical implications besides the one that I just went over. So there are being developed a number of more sophisticated techniques to pick up this mosaicism, and I won't go into detail of all of those, just to say that they are in development. They are looking at doing additional sampling sites. So again, we talked about you may not find things if you look in the blood, so we should look in skin samples in the cheek. But what if we could get even closer to the brain without doing a biopsy? Could we find some of these things in spinal fluid? There's also a way to go through the nasal mucosa to get quite close to the base of the brain. Would that be a better sampling technique? I think we're also going to be seeing this as having implications for parents and patients with epilepsy. If this type of mosaicism exists in their own cells, that may change the risk that they could have an affected child with epilepsy. I think there are some implications for treatment. There are some targeted medications that are being used for various epilepsy conditions, including everolimus for tuberous sclerosis. And if we could identify that the structural malformation was due to a particular change in a gene, could we target a medication and circumvent the need for surgery? Or what if surgery was not an option because of where the lesion was located? Could we be doing a better job at treating these structural epilepsies? And so there are a lot of really smart people working on this. They've actually combined forces into the Brain Somatic Mosaicism Network, which is funded by the National Institute of Mental Health to study this type of genetic brain disease across about 15 institutions across the United States, and not just for structural issues and epilepsy, but also for other psychiatric and neurologic diseases. Next slide. So the next thing we'll talk about is something called missing heritability. And this term was first coined in 2008 to describe the fact that although we had done this fantastic thing and sequenced the human genome, we had all these new technological advances, we hadn't really uncovered the genetic basis for both common diseases like high blood pressure and asthma and diabetes, or rare conditions like epilepsy. To say this in another way, people might say, well, you could also say it from the patient perspective. Why has all the genetic testing that my doctor has done for me, not yielded a result when a genetic diagnosis is suspected as the cause for epilepsy? And there are a lot of different complex and multifactorial reasons for that. But I would like to talk about some of those and some of the things that are being done to unravel some of those lingering questions. Next slide. So these are some of the considerations that make this such a challenging thing. So the first is something called phenotypic heterogeneity. So that essentially is the fancy way to say differences in clinical presentation. So take this gene that I've listed there, ATP1A1, having one copy of that gene with a variant may be associated with neuropathy, so tingling, numbness, and dysfunction in the hands and feet. But if you have two non-working copies of that gene, it can be associated with kidney disorders, intellectual disability, and seizures. Wow, what a big difference in the clinical presentation of those two things. And so why would we even think to look for a change in this gene in someone who has epilepsy if it's mostly involved in kidney disorders, right? And so that kind of disparity has made gene discovery and gene understanding a little bit more challenging. On the converse of that is genetic heterogeneity. So the fact that a disease can be the result of changes in a number of different genes. So if we look at various diseases like I've listed there, cystic fibrosis is a result of only one gene. That gene always causes cystic fibrosis, nothing else. If we look at tuberous sclerosis, that can be caused by changes in one of two genes, TSC1 or TSC2. But intellectual disability has been linked with changes in over 800 genes, and epilepsy with changes in over 400. So much more complicated to sort out what is responsible for what in those situations. Just one type of genetic or just one type of neurotransmitter within the brain, so NMDA, which is responsible for excitatory neurotransmission or part of excitatory neurotransmission, kind of our gas pedal in the analogy of a car, that receptor and that transmitter is coded for, and its related products are coded for by a number of different genes in gene families, some of which I've listed there. So we can't just look in one spot, we have to look in a variety of different places, and that makes it hard to figure things out. In addition, even if we identify a single gene like TSC1, there are lots of different genetic mechanisms which can cause disruption in that gene. So that gene can be completely deleted. There can be a single letter substitution in that gene that causes it not to fold or work properly. It can be, kind of turned around and stuck into a different part of the genome, which is called a structural variation. It can be, there can be a variation in a spot that tells the gene how to cut and splice together the various parts of the protein. And so all of these different mechanisms may have slightly different presentations in the patient, and they may require slightly different precision treatments to address how the gene has been disrupted. Next slide. The next thing is that a lot of these genetic mechanisms are unknown. So the human genome has about 20,000 genes, and even that estimate has actually varied because it's hard to know exactly where the gene starts and stops, and so things have been adjusted over the last 20 years. But even if we take that as a ballpark, we know that the major website that categorizes these genetic diseases, OMIM, or Online Mendelian Inheritance in Man, contains information on about 15,000 of those genes, but it doesn't really contain detailed information or human disease information. Some of it is very preliminary. About 8,000 of those 15,000 are associated with single gene disorders like tuberous sclerosis or cystic fibrosis or those types of things. And then there are about 3,000 syndromes that it catalogs that we don't know. We know that it presents the same, it looks the same, it acts the same, but we don't know why. We don't have a gene associated with it. We have done some studies, including this one that I was involved in called the Epilepsy Genetics Initiative, that actually looked at how much gene discovery is going on? What if we've done a genetic test for someone, whole exome sequencing in specific, and then we look back at it a couple of years later? Has enough gene discovery happened that we would be able to find answers that we didn't know before? And the answer is yes. So if we look at an annual basis, we can discover about 6% a new diagnosis for those patients that was unknown at the time of the initial test. Next slide. Okay, sorry, I wasn't sure if we had changed yet. Okay, non-coding regions. So this is an example of kind of how this test works. So most commonly, what's done is whole exome sequencing, and that looks at the main information carrying portion of our genome. So this is where it contains the information about how to assemble proteins that then go on to have a specific job in our body. And those are called exons. I liken this to kind of a train with a boxcar. So the exons are the actual train cars, and then the introns are what we would say are the linkers in between those boxcars. And when the body makes a protein, it gets a signal to cut at the end of those linker proteins or linker DNA segments and push all the boxcars together to make the protein. So the exome is where most of the money lives. It contains about 3/4 of what we know about human genetic disease. But in the grand scheme of things, exons are only about 2% of our genetic material. The remainder of that genetic material is the genome. And that contains a lot of regulatory elements that, in addition to those introns, it tells our body when to turn things on, when to turn things off. If there's a higher level of this, then you should turn on this protein, or you should do this only in these cells. A lot of things that kind of help execute the plan of the body over time. And many of the genetic epilepsies that we know about do have variations in intronic regions associated with those genes. And I've listed some of the ones that we've been reported, that have been reported so far. And so this may affect how the gene is cut and put together, how the gene is turned on, how the body is told to start producing this protein, etc. And then another recent discovery has been a genetic association with a syndrome called benign adult familial myoclonic epilepsy. And actually, they found that repeat expansions, so chunks of just this same genetic code repeated over and over and over and over, in introns of a number of different genes, produce this similar clinical picture in patients. And I think that's important because most of our exomic technology for whole exome sequencing is not able to detect triplet repeat or repeat expansions based on how the testing is performed. So this was a really recent discovery because we had been doing whole genome that we identified these areas in between our genetic material that contain important information that can affect disease presentation. Next slide. The last thing is or not last, but the next thing would be technological advances. So again, exome sequencing is not able to detect these complex structural rearrangements. So again, when genes get kind of cut up or turned around and put into a different segment of the DNA or smushed together with another gene, those things are called structural variations. And up till recently, our testing was not very good at picking those up. We also have better databases of human variation, right? So all of us are slightly different from each other. If we're going to quantify that, we have about 3 million variations in our genetic sequence between any two given people. And because the population has expanded so much so recently, those variations are probably relatively unique and have not spread through the population. They may be very rare. And they may not be linked clearly to disease, which is what a lot of our previous genetic testing has revolved around. And so we need to have a big database to understand, is something that we see in a genetic test for a patient just part of normal human variation, or is it something that can be linked to a disease like epilepsy? Unfortunately, many of our reference genomes do not contain a lot of information about underrepresented populations. So people of African origin, of Asian origin, Native American patients, and that makes it more complicated to sort out whether those genetic variants that we discover in those patients are significant or not. The reference genome that most labs use to analyze their genetic test results actually came from about 13 people of European ancestry, which is clearly not representative of the breadth of human variation. So it's hopeful that because the technology has advanced so far and we are doing this testing more and more often and the cost is decreasing further and further over time, that more people will be able to make use of this technology, and then we will understand more about human variation and get better technology that can get more answers for people. Next slide. So I just put here a few studies that I thought were really illustrative of some of these points. So the first was done and reported in neurology just this year. And these were patients who had had exome sequencing, so our kind of best clinical test at the time, that did not find an answer for them. And they went on to have whole genome sequencing. So looking at the remainder of that genetic material containing all those regulatory pieces of information, as well as those linkers in between the boxcars. This was about 30 patients, all of whom had severe epilepsy syndromes of childhood onset. And they found 5 new genetic diagnoses that were not really understood at the time of their initial testing. 3 patients had some of those structural rearrangements that we talked about that were not detectable by the initial technology. 10 patients had had a very large panel, which is another common way to do genetic testing. But obviously, as soon as it's published and starts to be used, it's out of date because we've already discovered another gene that's not contained on that panel. And so that was the case for about 10 patients. So overall, the yield of this particular study was about 63% of the 30 patients got a genetic diagnosis, but most of them would probably have been detected with better technology or repeat analysis of their initial data. Recently, we saw very similar things in the Child Neurology Foundation, who sponsored a pilot program in which 20 patients from across the country, 5 from a number of different pediatric academic centers, were given free access to whole genome sequencing through this pilot program. And the yield for this was also about 25%, so 1 patient from every center. Our case that we submitted that was able to get a diagnosis was one of those complex structural rearrangements. In my experience in clinic, when we have sent genome, most of the results that we have gotten have also been discoverable by exome, but just not known at the time that the patient's testing was sent. So I think this is still kind of an evolving answer about how whole genome sequencing will play into the diagnostic evaluation of genetic epilepsies. Next slide. The next potential explaining factor for this missing heritability is that perhaps epilepsy is polygenic, meaning that it may not be a single gene that causes epilepsy in a particular patient, but a combination of genes. And you'll see up there in the right-hand corner the kind of pictorial representation of that. So if we add a number of genes together and their potential effects or their potential spellings, over time that begins to form a normal curve. And if we think of that normal curve on the picture next to it as, say, something about a risk for epilepsy or liability towards epilepsy, it may be that as a number of those different variations combine, at some point you cross a threshold, and that is when spontaneous seizures occur and epilepsy is diagnosed, like a seizure threshold. So there are some cases where this has been shown in 2 genes, so 2 genes within the same pathway combined together have produced a similar syndrome in patients. The fancy medical term for that is digenic and biallelic, meaning 2 genes seen on different alleles, which is the copies that we get from our mom and our dad. A larger study actually looked at polygenic risk scores. So this was a large study of about 2,000 patients with epilepsy compared to 20,000 controls or people without epilepsy. And when they looked at the potential differences in how many genetic variations those patients had in epilepsy genes, they found that that maybe accounted for about 3% of the difference between those 2 populations. So not a lot, but some. There's been another study that I cited down there at the bottom from the Epilepsy 25 Collaborative and Sam Berkovic in Australia, that when they studied a cohort again of 9,000 patients with epilepsy and looked at them, they found an excess of what are called rare deleterious variants. These are variants that cause the protein to stop being made, to be degraded by the body, and to be non-functional. Those are usually particularly damaging to that protein. Unfortunately, no gene rose to statistical significance from a scientific perspective, such that we may need to combine into even more teamwork and large sample sizes to get the answers that we need, because we are looking from that graph on the left for things in the very lower left-hand corner, rare variation in the population with a small effect size, meaning it has to be seen in combination with a number of other genes in order to produce the symptom. And those things are very difficult to identify and take large numbers of patients. Next slide. So there was a study done in 2018 funded by the ILAE, the International League Against Epilepsy Consortium on Complex Epilepsy. This is a mechanism that we had used previously to try and identify the genetic causes of epilepsy and try to identify some of these smaller effect size genes called GWAS, or Genome-Wide Association Study. And this actually did examine a larger group of patients, 15,000 cases and almost 30,000 controls. For some perspective, cases that have, or studies that have done a similar thing for schizophrenia involve 37,000 cases, so about double what we had. They identified a number of potential areas that variation was felt to be significant, and those did overlap with some genes that we already know about in epilepsy. And they did some additional analyses, but they really weren't able to, get a lot of information to move the field forward. So a larger study is currently underway using a larger number of patients, about 30,000 patients with epilepsy, again, looking for some of these polygenic contributors to undiagnosed causes for epilepsy. Next slide. Another potential contributor to missing heritability is transcriptional variation. So when we talked about cutting those boxcars and smushing them together, we know many of the places that those genes are cut and pushed together. They're called canonical splice sites, meaning that's the most common form of the gene. It's expressed in most of the tissues in the body. It's what the body makes 90% of the time. However, there are other ways that that gene can be cut and pasted together, as you can see in the picture there. And is it possible that that is what is occurring in the brain, and that changes in some of those other splice sites that are less commonly used by the body is what is responsible for the epilepsy presentation? And so they have done some additional testing. We have some new technology that's able to highlight some of these alternative splice sites, which look a little bit different, and supplement interpretation of genetic testing. And when they did that, and this is not quite published yet, and it's from a testing company, but when they did that in 88 samples, they learned about 100,000 new gene transcripts, and looked at them but based on protein expression to see, did that actually get translated to functional proteins within the body? And it did. And if we put those two things together, if we put together our traditional interpretation and sequencing interpretation with this new information about alternative splice sites, we may be able to increase the yield of our testing by up to 30%, which was what those writers anticipated. That has also been something that has been shown by the Epilepsy Genetics Initiative. They found at least a few cases of alternate splicing in SCN8A that was responsible for presentation of genetic epilepsies in that cohort. There's another potential nugget here is poison exons. So these are exons that are included into the final transcript and signal the body to degrade that particular protein. And these are thought to play a role in how the body regulates different levels of gene expression and kind of keeps them at the precise balance that it needs. And this type of poison exon may be more common in our brain because we need to have such precise regulation of gene expression there. And it has been actually implicated in a number of neurodevelopmental genetic disorders, including SCN1A, which will become important in our discussion about genetic therapies coming up. Next slide. So this is just a brief section about modifier genes. Next slide. So these are the fact that if you think about it, even if you have a monogenic disorder like SCN1A or Dravet syndrome, that's not the only gene that that person has, right? We are composed of 20,000 genes. And so could changes in other genes affect the presentation of the primary disorder? And that actually has been shown in patients from Japan with epilepsy. And those patients had an excess in, again, ultra rare and damaging variations in other epilepsy genes in a higher number than patients without epilepsy. And so is it fair to say that all epilepsies, even known single gene epilepsies, are oligogenic, meaning formed by the active and interaction of several genes rather than just one? And I think that very well may be the case. For SCN1A, that has kind of made the most progress in identifying some of these modifier genes. And they've found that patients may have one that accounts for some variability or several. And there have been actually a number of mouse models. So when we study mice, they are very inbred. We use the same mice, for lots of different things. And so they found that the expression of the Dravet model was different depending on which strain of mouse was used. And so they've been able to kind of replicate that and use that to zero in on what were the differences that accounted for that variability in mice. Unfortunately, even though we know that there are modifier genes and we have a few of them identified, we are not yet at the point where we can say, okay, this is your primary diagnosis of Dravet syndrome, and because you have variants in these several other genes, we think that your Dravet is going to be more serious or less serious. We can't really do that at this time. Next slide. I think this was an exciting discovery for me in preparing for this talk, is that some of those modifier genes may actually be lurking in very unexpected places. So on the left, I've put a cartoon here about how the body translates our genetic code into proteins, right? So it has the messenger RNA at the bottom there with all the letters and the little, rungs or sprockets. And then the amino acid is brought in by something called tRNA or transfer RNA. RNA, and that is added on to the protein chain in something called the ribosome, or an organ within the cell. And so again, using these inbred mice, they had a pathogenic variant in an inhibitory gene that's related to epilepsy. So the brake pedal, if we think about our analogy. And the mutation in this transfer RNA kind of decreased its efficiency and its ability to participate in this process of gene expression. And because it decreased the translational efficiency, it again affected a number of genes, including our friend mTOR, who we heard about in the first part of the discussion, involved in structural epilepsy. And so the thought is, well, we don't even have to alter the foundational mutation in this patient in the GABA gene. What if we could just upregulate some of the other tRNAs that our body uses and circumvent this one that's been made less efficient? Could that help? And in fact, it did actually improve the resistance of those mice to experimentally generated seizures, which is really exciting. Next slide. Okay, so this is the last section. I hope that I will be able to get through it in the time remaining, but since Chelsea is the next presenter, we will just maybe run into her talk a little bit, and she can tell the person behind her that we had some technical difficulties. So we'll give it our best.
My lecture, I think, is a little short of an error, a little short of an hour.
Great.
So take your time.
Okay. Great. So, so this will be a discussion about precision medicine and gene-based therapies. And I just want to give a shout out to our other genetic counselor, Melissa Gibbons, who gave me a number of these slides so that I could present them to you today. Next slide. All right, so again, we've talked about this a number of different times. How does a gene become a protein, right? How does a bill become a law? I guess I'm showing my age there, but we start out with DNA, and then that is transcribed, is the name of the process, into RNA. Then it's spliced, and those boxcars are pushed together to make messenger RNA that comes out of the nucleus and goes into the body of the cell, and is taken to the ribosome, where it is translated and made into a chain of amino acids. Those amino acids then fold themselves into a particular conformation that makes a functional protein. So how could we alter that in gene-based therapies? So we could either stop something, we could make something more active, we could edit it and cut out the part that has the variation in it, or we could replace that gene altogether. And all of these mechanisms and a number of other different options are being explored, and that's what the next slides will talk about. Next slide. So these are the three things that I'm going to kind of cover in some detail today. Antisense oligonucleotides, which I will refer to as ASOs, gene transfer therapy, and then CRISPR, which is also broadly used in medical research nowadays. Next slide. So antisense oligonucleotides, or ASOs, they are very short, artificially created, synthetic, single-stranded oligodeoxynucleotides that can come in and alter our RNA and change protein expression. Typically, they are between 8 and 20 nucleotides in length, and they bind to a specific sequence of RNA through, A to T, G to C, those types of things. That's Watson-Crick base pairing. And they therefore regulate the expression of genes. And this kind of therapy is not actually science fiction. The first ASO called, well, I don't know how to pronounce that, but fomaversin was approved by the FDA in 1998 to treat CMV retinitis in patients with HIV/AIDS, and was given by intraocular injection, so injection into the eye. Additionally, two further ASOs were approved by the FDA in 2016 for the treatment of Duchenne muscular dystrophy and spinal muscular atrophy, very severe neuromuscular diseases of childhood. Unfortunately, ASOs often are only able to prevent a disease from getting worse. They may not be able to undo some damage that has already been done, which is why a lot of these discussions will revolve around figuring out what time these therapies need to be administered, in what window, and that may differ for various diseases. And we want to certainly, for progressive things, administer them before the disease has become too severe. Next slide. So, what are some of our techniques? So we can augment the expression of a gene, and that's usually used to treat diseases that are caused by a mutation that stops the gene from producing a functional protein product. We can inhibit the expression of genes, and that is useful when the product of the mutated gene inhibits the expression of another gene in the body or interferes with the product of another gene in the body, and so we want to remove it from the picture. We can introduce new genes, and that can be done either into the body by the delivery of a gene packaged with inside a viral vector, typically adenovirus, to get to the target cell, or ex vivo, where patient cells are extracted from their body. They are altered with this technology and then infused back to the body. And you may have heard of that in CAR T therapy, which is being used as immunomodulatory therapy for cancer in many situations, especially lymphoma. Unfortunately, delivery to the nervous is quite challenging because of something called the blood-brain barrier, and the therapy has to be delivered close to the target so that it doesn't get degraded by the body. And so that means things like injection into the substance of the brain, into the spinal canal through a lumbar puncture, into the ventricular system through a shunt or an indwelling device. Although people are actually trying to work at some less invasive options. So again, through that nose, which is leads right to the brain through the sinuses. And they're looking at that for some conditions. Next slide. So what are some of the challenges of gene transfer therapy? So we have to get the gene to the right spot, and then we have to have a mechanism to be able to turn it on and make the body produce that copy. We have to avoid the immune system, so the body attacks anything that it sees as foreign, and we don't want it to degrade the product that we're giving it or the gene that we're giving it before it even gets there and has a chance to work. We have to make sure that the gene that we're delivering doesn't get put in the wrong spot and disrupt the function of other genes that we do want to keep working. We also want to make sure that those vectors that we're using to deliver the gene don't cause a mutation where they are inserted. And finally, once we turn it on, we typically don't have a way to turn it off. And so we want to make sure that it's not going to cause any other problems. Next slide. So this next technology that I'm going to talk about is one of the main technologies involved in delivering gene therapy, and it's called Crispr. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. That's a lot to say. Basically, it is an RNA-guided gene editing tool. It's typically paired with something called Cas9. So you'll often hear it referred to as CRISPR-Cas9 because they usually work together. And the Cas9 is the targeting protein that helps it to remove make the splice and put in the new sequence. This basically not only has been used for gene therapy, but has kind of revolutionized the science that we use to learn about genetic disorders by enabling the creation of a number of animal models and changing cells and looking at the effects of things in different cell lines. It's been kind of one of the greatest inventions of our century. So I've put kind of a little pictorial representation of what this protein actually does. As you can see, it's attached to a custom designed sequence. It can go in and break apart the target DNA and insert the RNA copy that we want into that. The biggest change about it is it can disrupt DNA and break it apart in a single chain without having to make a double cut on both strands, which is what has caused problems with gene therapy in previous iterations that we have worked with. Next slide. So what are the challenges with this? And just to say also, this is not science fiction either. There are some trials currently ongoing for gene therapy with this technology. The one that I listed there first was published in the New England Journal of Medicine in this year to treat a hereditary lung disease, and they've infused that therapy and are monitoring those patients. There's another trial that has been done for beta thalassemia and sickle cell, both blood diseases, designed to increase the production of fetal hemoglobin, which is usually not affected by the same mutation and has the same problems. So upregulating another gene product to reduce the symptoms of the disease. But what are the challenges if we want to do this? So the first is that we need to identify an efficient and safe delivery system. Most of the ones that we have available are viral vectors, as we talked about, but there are some non-viral physical methods. So being able to poke holes in the cell and deliver the product through that way, using lipid molecules that contain a copy of the gene that we want. I don't exactly know what gold nanoparticles have to do with things, but that was on Melissa's slide too. We also need to avoid these off-target effects, so disrupting other genes, changing signals that are surrounding cell death and cell regulation. We don't want them to insert someplace else and cause a deletion. And then when it cuts this single strand, it leaves a little piece of this artificial DNA. And we don't really know what having a bunch of that around is going to do functionally for people. Will it get degraded by the body, or will there be problems from it? Next slide. What are some additional challenges for neurodevelopmental disorders? So again, we talked about this timing piece of things, right? It may need to be delivered, whoopsies, oh, okay, in a critical period before brains have matured, and that critical period may be different based on different disorders, and we will need to understand how that gene functions both in childhood as well as all the way back to fetal development in order to figure out when that critical window is. Unfortunately, a number of the genes associated with neurodevelopmental disorders have similar symptoms when there is an excess of protein. And so we call this the Goldilocks phenomenon. You can't have too little and you can't have too much. You have to have exactly the right amount. We are also dealing with regulatory type genes. So genes that don't just have one function, one defined function in the body, but may regulate a number of other gene products. And so if we change their function, how is that going to affect all the other things downstream of that? And I've listed some of those potential proteins there. And do we need to have a differential expression? So do we need to have a different amount of this protein in different cells? Does that depend on what type of cells or what time we are functioning in that critical window? Not clear yet. Next slide. So now we're going to get into some big science. So I put some mice here, and my discussions with our fellows, no talk is ever complete without mice. So this is the mice. So I'm going to talk about a few diseases, but I want to caution you that mice are not people. So many researchers have said, if I could cure, I've cured Parkinson's 10 times in the mouse, but not in people. So just because this may have some effect in mice does not mean that it is present, it's going to work in people. And all of these therapies are in their very early stages, looking at animal models which may not have all of the symptoms of the disease that exist in people. Next slide. So the first condition we'll talk about is DRAVET, and since this is one of the first epilepsy genes identified, it kind of has made the most progress, and so there are a number of slides about SCN1A. And so one of the first technologies that they're developing is something called TANGO, and I've listed the acronym there. This is an ASO, so an antisense oligonucleotide delivered in a lipid nanoparticle to limit the immune system's ability to target it and destroy it before it gets there. It blocks inclusion of a poison exon, which we talked about before, in otherwise functional copies of SCN1A. It's again a normal part of transcription that then the body would degrade those copies, but instead this TANGO, this gene therapy, is saying, oh, don't do that. We need that copy of SCN1A because one of our other copies isn't working. So don't destroy that one. We're going to cut out the poison exon, and then it's going to work, and it should be fine. Again, the dysfunctional copy is still made and still degraded by the body, but this extra copy is not. This would need to be delivered through the spinal fluid. And in a mouse model, some preclinical studies have shown a decreased mortality for the mice and decreased seizures. And early clinical trials of this particular gene therapy began in 2020. And that is a graphic from the website of the pharmaceutical company sponsoring those clinical trials. Next slide. Unfortunately, the SCN1A gene is too large to be packaged inside one of those viral vectors. So a different therapy, which again I took from the Encoded Therapeutics website, delivers a promoter factor to increase the production of SCN1A. So it again attaches to one of those regulatory signals and says, make more of this protein because one of the copies isn't working. The vector that is being used is the same one that's used in the already approved SMA medications, which have been around since 2016. Again, functional copies are still degraded by the body. It does not integrate into the body's genome, which is an attractive feature. Instead, it forms this circular thing called an episome. And it's specifically targeted to inhibitory interneurons, which is a specific population of brain cells where SCN1A is predominantly expressed. So that's an additional beneficial factor. Preclinical studies have also shown improved survival and decreased seizures in a mouse model. This is given again by injection into the ventricular system of the brain a single time. And a clinical trial of this particular gene therapy is anticipated probably next year based on what we've heard so far. Next slide. A couple other options are being developed as well. So this is a kind of, again, upregulating production of SCN1A by adjusting the body's normal processes, overexpressing some of that transfer RNA to prevent degradation and allow more protein to be expressed. Suppressing codons that contain stop or nonsense mutation by giving transfer RNAs that correct that amino acid sequence. The preclinical data for this particular technology has been shown in cells only and is moving towards animal models and is again delivered by a viral vector. Next slide. So additional things that are happening, they are trying to design larger vectors so that they can put the whole gene in there. Some people are looking at splitting the gene into two parts and putting one part in each vector, and then the body would assemble them together into a full copy. And they're also doing some other things to activate other regulatory regions of SCN1A, which I've indicated there. And there are some references at the bottom if anyone is interested at reading more about those. Next slide. So Rett syndrome is also making progress towards gene-based therapies. This is again done in an animal model where they infused into the vein of the mouse's tail a vector delivering a functional copy of the gene to the mice. Now these mice had no functional copies of the gene, as opposed to human disease in which the mice have one functional copy of the gene. But delivering this gene improved the motor and behavioral symptoms of the mice and slowed the disease progression. However, again, we get into that Goldilocks phenomenon that delivering a higher dose of this gene to the mice was associated with toxicity and similar symptoms as the affected mice. So people have been working on that by trying to deliver a copy of the gene that maybe isn't produced as efficiently, and so maybe it's just a gentler increase in the gene product. And so that is a very interesting development. They're also looking at trying to use IV injections rather than intrathecal injections for people, but they've had some differences in how the different mice strains have responded to some of that testing. Next slide. Angelman syndrome has done some research as well with injection right into the hippocampus, the memory centers of the brain, a viral vector containing a copy of the gene, where it has been tested. Oh, I was supposed to put the reference in there, but I guess I forgot to do that. I'm very sorry. So they're still working on that in mice. It's also considered a disorder of imprinting. So basically, we get 2 copies of the gene, right? 1 copy from our mom and 1 copy from our dad. And in a patient, typically the paternal copy, the copy from dad, is silenced, and therefore the only functional copy is the copy that you get from mom. And if mom's copy is not working well or contains some sort of, or is deleted or so forth, then that's when the disease is symptomatic. So the techniques that we use to kind of get around that are to give, again, an ASO to help silence what makes dad's copy not expressed. And so they tested this in mice, and it did work. It was able to improve some of the learning symptoms, but not some of the other issues associated with this mouse model. Improvements were seen when the injections were performed at various points in time. So when the mice were injected in utero, as well as when they were injected several weeks after they were born, which is encouraging news. Next slide. Fragile X syndrome, which is the most common monogenic cause of autism and intellectual disability and frequently associated with seizures, has also been doing some gene therapy trials. And delivery of the gene by vector improved symptoms of mice, again, without any functional copies, so different than the human disease. They've also used the CRISPR-Cas9 technology that we've talked about to excise the triplet repeat expansion that is responsible for the disease in human pluripotent stem cells. And other types of therapies have also been designed to balance some of the downstream effects of not having a functional FMRP protein. Again, this is one of those regulatory genes that controls a lot of other things within the nervous system. And so kind of trying to get that balance restored without actually giving a new copy of the FMR1 gene is what this group is looking at. Next slide. Tuberous sclerosis has also been doing some things. They did a vector with TSC1, and they were able to extend survival in mice when they injected that gene in infancy. And SCN8A, epileptic encephalopathy, is also using ASO technology, which has been able to delay seizure onset and prolong survival in a mouse model. And interestingly, again, as these modifier genes that we talked about, some of these changes to SCN8A also were able to help SCN1A mice who mimic Dravet syndrome. So that's encouraging as well. Next slide. Yay! Okay, that's the last. So I only went about 10 minutes over, and I will then turn it over to Chelsea to begin her discussion about the ketogenic diet. I know that was a lot of information. I thought there was going to be a thing for questions, but I don't know if that got removed from the website. But I hope everyone has enjoyed the talk and learned some things about genes and gene therapy.